Tonight's lecture is over Chapter 6, and it deals with the photosynthesis part of this chapter. So we were going over this in the last lecture concerning cellular respiration, but just for a quick review, a metabolic pathway is a series of chemical reactions that takes a starting molecule and modifies it step by step through a series of metabolic intermediates, eventually yielding a final product. Anabolism is the process of synthesizing something or building something, like an anabolic steroid builds muscle, and degradation or catabolism is the process of breaking something apart. And chemical reactions of metabolic pathways do not take place under their own. Each step is catalyzed by an enzyme, and we can see examples of both an anabolic and catabolic pathway in the cartoon on this slide. So this is more review because we did discuss energy and thermodynamics in the working cell chapter. But thermodynamics refers to the study of energy and energy transfer involving physical matter. The matter relevant to a particular case of energy transferred is called a system, and everything outside of that matter is called the surroundings. And in an open system, energy can be exchanged with its surroundings, and in a closed system, energy cannot be exchanged with its surroundings. So there's two laws for thermodynamics. The first law states that energy cannot be created or destroyed. It can only be transformed into something else. So in other words, there has always been and always will be exactly the same amount of energy in the universe, but energy can exist in many different forms. And it's relevant to our discussion of photosynthesis because plants perform one of the most biologically useful energy transactions on Earth. They convert the energy of the sunlight to the chemical energy stored within organic molecules, and this is critical for life. And the second law of thermodynamics states that in any energy transformation, there is always some energy that is lost in a form that is unusable. So an example of that would be, for instance, in your car. When you turn on your car, you're converting energy into another form to do work, and some of that energy is lost as heat. And this also happens during cellular metabolic reactions. And recall from the working cell chapter, we also talked about the types of energy. Kinetic energy is the energy of motion. So when the diver is climbing up a ladder to get on the diving board, that is an example of kinetic energy. As the diver walks across the diving board to the edge of the diving board where the diver will dive off, that's another form of kinetic energy. But then when the diver stops at the edge of the diving board, there's potential energy, and that's the energy associated with location and structure of matter. For instance, a diver on a raised platform. There is potential energy stored within the bonds of all food molecules we eat, which is eventually harnessed for use. And because these bonds can release energy when broken, the type of potential energy that exists within chemical bonds and is released when these bonds are broken is called chemical energy. So chemical energy is essentially a form of potential energy. And there's also types of chemical reactions. So an exergonic reaction has a negative change in free energy that releases free energy. So if the energy is raised during a chemical reaction, then the change in free energy is signified as delta G, and this will be a negative number. A negative change in free energy also means that the products of the reaction have less free energy than the reactants because they release some free energy during the reaction. And on the other hand, an endergonic reaction will have a positive change in free energy. And this is because they require the addition of free energy. So if a chemical reaction absorbs energy rather than releasing energy on balance, then the delta G for that reaction will be a positive value. And in this case, the products have more free energy than the reactants. So how does this relate to cells? Well, a living cell cannot store significant amounts of free energy. Excess free energy would result in an increase of heat in the cell, which would result in excessive thermal motion that could damage and destroy the cell. So a cell must be able to handle that energy in a way that enables the cell to store energy safely and release it for use only as needed. Cells use ATP as the main energy storage molecule, and when ATP is broken down, energy is released. When ATP is built, energy is stored. So by that statement, we can extrapolate that when a chemical bond is broken, energy is released. And when a chemical bond is created, energy is stored. So breaking a bond releases energy, creating a bond absorbs energy or stores energy. So that brings us to photosynthesis, which is a process whereby plants, algae, which are protists, and certain bacteria transform light energy into chemical energy using carbon dioxide and water as the starting materials. The chemical energy produced via photosynthesis is stored in the bonds of sugar molecules. So remember, organisms that generate their own organic matter from inorganic ingredients are called autotrophs. Plants and other organisms that do this by photosynthesis are called photosynthetic autotrophs and are the producers for most ecosystems. Moving on to light energy. Sunlight is a type of energy called radiation or electromagnetic energy. Electromagnetic energy travels through space as rhythmic waves analogous to the ripples that are made by a pebble dropped into a pond. The distance between the crests of two adjacent waves is called a wavelength. The full range of radiation from the very short wavelengths of gamma rays to the very long wavelengths of radio signals is called the electromagnetic spectrum. Visible light is only a small fraction of the spectrum. It consists of the wavelengths that our eyes see as different colors. And we can see in the cartoon on this slide that frequencies with shorter wavelengths have more energy than frequencies with longer wavelengths. So therefore, violet has more energy than red. So when sunlight shines on a pigmented material, certain wavelengths or colors of the visible light are absorbed and disappear from the light that is reflected by the material. For example, we see a pair of blue jeans as blue because the pigments in the fabric absorb the other colors, leaving only light in the blue part of the spectrum to be reflected from the fabric to our eyes. Moving on, photosynthesis in plants and algae occurs within the light-absorbing organelles called chloroplasts. All green parts of a plant have chloroplasts and can carry out photosynthesis. In most plants, however, the leaves have the most chloroplasts and therefore are the major locations of photosynthesis. Their green color is from chlorophyll, a light-absorbing pigment in the chloroplasts that plays a central role in converting solar energy to chemical energy. Chloroplasts are concentrated in the interior of cell leaves. Carbon dioxide enters and oxygen exits by the ways of tiny pores called stomata. In addition to carbon dioxide, photosynthesis requires water, which is absorbed by the plant's roots and transported to the leaves, where it is carried to the photosynthetic cells. Membranes within the chloroplast form the framework where many of the reactions of photosynthesis occur. Like a mitochondrion, a chloroplast has a double membrane envelope. The chloroplast's inner membrane encloses a compartment filled with stroma, a thick fluid. Suspended in the stroma are interconnected membranous sacs called thylakoids. The thylakoids are concentrated in stacks called grana. The chlorophyll molecules that capture light energy are built into the thylakoid membranes. The structure of a chloroplast, with its stacks of discs, aids its function by providing a large surface area for the reactions of photosynthesis. There are two types of photosystems that cooperate in light reactions. Photons excite electrons in chlorophyll of the water-splitting photosystem. The photons are trapped by the primary electron acceptor. The water-splitting photosystem replaces its light-excited electrons by extracting electrons from water. This is the step that releases oxygen during photosynthesis. Energized electrons from the water-splitting photosystem pass down an electron transport chain to the NADPH-producing photosystem. The chloroplast uses the energy released by this electron fall to make ATP. The NADPH-producing photosystem transfers its light-excited electrons to NADP+, reducing it to NADPH. And this slide just shows two cartoons of both photosystems. So we can see on the left-hand side we have photosystem 2, or the water-splitting photosystem. And then on the right-hand side we have photosystem 1, or the NADPH-producing photosystem. So don't get confused. In the photosynthetic reactions, photosystem 2 occurs first, and it leads to photosystem 1. The reason for this nomenclature is because photosystem 1 was discovered and named first. Then photosystem 2 was discovered. Photosystem 1 is dependent upon the electrons that are liberated by photosystem 2. So that's why photosystem 2 occurs first and photosystem 1 occurs second, and that is the reason behind their nomenclature or their naming system. Moving on, the Calvin cycle functions like a sugar factory within the stroma of a chloroplast. It is called a cycle because, like the citric acid cycle in cellular respiration, the starting material is regenerated with each turn of the cycle. And with each turn of the cycle, there are chemical inputs and outputs. The inputs are CO2 from the air, as well as ATP and NADPH produced by the light reactions. Using carbon from carbon dioxide, energy from ATP, and high-energy electrons from NADPH, the Calvin cycle constructs an energy-rich sugar molecule called glyceraldehyde 3-phosphate, or G3P. The plant cell can then use G3P as the raw material to make the glucose and other organic compounds such as cellulose and starch. The Calvin cycle occurs in three basic stages: fixation, reduction, and regeneration. Furthermore, it takes 6 turns of the Calvin cycle to fix a 6-carbon atom from CO2. These 6 turns require energy input from 12 ATP molecules and 12 NADPH molecules in the reduction step and 6 ATP molecules in the regeneration step. And on this next slide, we simply see a cartoon of the Calvin cycle. So initially from the air, we have an input of CO2. And then at the first step, an enzyme adds each carbon dioxide molecule to a 5-carbon sugar called RUBP. The resulting molecule breaks into 2 3-carbon molecules. Then at the second step, using energy from ATP and NADPH produced by the light reactions, enzymes convert each 3-carbon molecule to the 3-carbon sugar G3P. Thirdly, for every 3 molecules of carbon dioxide that enter the cycle, the net output is 1 G3P sugar. The other G3P sugars continue in the cycle. And at the 4th stage of the Calvin cycle, using energy from ATP, enzymes rearrange the remaining G3P sugars to regenerate RUBP. So to summarize this cartoon, we could say with the energy of ATP and the electrons from NADPH, the carbons from CO2 are used to make a 3-carbon sugar called G3P. And that brings us to our summary of photosynthesis. So now that you've heard the lecture and read the material, you should know what the two basic types of metabolic pathways are, and which produces energy and which consumes it. You should be able to define an open versus a closed system. You should be able to relate the first and second laws of thermodynamics to biological systems. You should know what the two main types of energy are. You should be able to define types of reactions in terms of free energy. You should know how the wavelength of light relates to its energy. You should be able to describe the basic steps of the light-dependent reactions of photosynthesis. And you should also know what occurs during the Calvin cycle or the dark reaction.